everyone, today I'm going to talk about protein biosynthesis. In order to understand this procedure better, let's first see some basic structures these molecules are involved in this procedure. Okay, let's first see the structure of a protein. We want to understand how we get a protein, and first let's see the basic structure of a protein. A protein is made of amino acid. It's like a chain of amino acid. Just like the picture showing here, it's a pearl of necklace made of pearls, and we can think about protein as also a chain or necklace made of amino acid, just like one pearl. Okay. However, I have to introduce you other structures of the of some molecules. Molecular is a thing, a small thing in our body or somewhere. I have to introduce you some molecular structures of other very important things involved in this procedure. Now let's first see the structure of DNA. And the DNA is, we can see in this picture, has two strings, and the two strings has curled together. Just think about a dip in your coat. Oh, sorry, I don't have a dip. Here is a dip, and we can think DNA as a zip. In the structure of DNA as a zip. And next, we have to also to understand the structure of mRNA. We can see mRNA only have one strings compared with DNA. So we can say mRNA is only one string from a zip. Also, mRNA and DNA are made of different nuclears. mRNA and DNA are made of different branches in this zip. This one. It's a little different, but you don't need to understand this. And now let's see the structure of tRNA. tRNA is a very important molecular. It is a very important thing involved in this procedure. In order to understand the structure of mRNA, I need to show you 3 points. The first point is mRNA is also like a zip, but we can see only one chain of a zip, and also we can see it's very short in this part. And the second point is mRNA is, let's see, if mRNA is come from one side of the zip, then tRNA will come from the other side of the zip. So we can know these two RNAs can combine with each other very tightly. And the third point is mRNA carry an amino acid. Remember I have said the protein is made of amino acid, right? Okay. So we can see the structure of mRNA. Okay, we have almost there. We have just mentioned the amino acid is just like a pearl in the protein chain, a necklace you can think. Okay, the last structure is the whole structure of a cell. The cell is made of 2 parts. We can see this part in the nuclear, and outside the nuclear, we say it's the cytoplasm. And also, I have to mention DNA exists in the nuclear, and tRNA exists in the cytoplasm. Here's the general look. Do you think it's beautiful?
Yeah, as beautiful as you.
Thank you, boy. Okay, now let's come to the point today. Can I use it? Now our today's topic is about protein biosynthesis. In order to get a protein, in fact, we already have, I have shown you in this picture, we already have DNA, and we already have tRNA, and we already have amino acid, I can say it, in our cell. So, what's the problem in synthesis of protein? I will show you. To get the right protein, we have protein have different sizes, different shapes. Also, we have 20 kinds of amino acids. So what do I mean? we want to, let's say, we want to have a protein with 10 amino acid. So we have a lot of combines to get this kind of structure, right? I mean, the first amino acid could be the A protein, A amino acid, B amino acid, and C amino acid. We have 20 amino acid. So the first amino acid can be any of the 20 amino acid, and the second also have this choice.
So you mean if 20 by 20 like
no,
a lot of bananas?
It should be. I mean, you will
20 multiple 20 multiple 20 y.
It should be like
Yeah.
this, right?
Yeah.
So the problem is how can we decide the sequence of amino acid in a protein? What's your problem?
I think it's 10
20?
times 20.
Yeah.
20
times 20.
20.
Oh, come on, boy, don't argue with me. The first one can be A, B, C, D, E, so we have 20 choice.
Yeah.
And the second we have 20 choice.
Yes.
Oh.
So it's 20.
It's 20. Whatever. It's not the main thing about today. I think you can understand this.
Okay.
Yeah.
I think you can understand. Now our problem is how can we decide the sequence, right?
Yes.
Okay, I will show you here. We have DNA. And we want DNA to determine the amino acid sequence. That's what I will show you next, the basic procedure. Okay, now let's come to the point today. In order to get a protein, we have two basic procedures. The first procedure is called transcription. And this procedure happened in the nuclear. As I mentioned before, DNA exists in nuclear, right?
Yeah.
So in this procedure, we basically to get mRNA. And in this procedure, two DNA strings will departure with each other, and the ranchit, I mean, this 1, 2, 3 from these things, is not amino acid, it's other kind of moleculars. And this ranchit will add one by one based on one string of the DNA. It's kind of very strict thing to decide the sequence of the mRNA first. And this string is called a template string. And after this ranchit will also hand in hand together. We can think it hand in hand together to make a stable string of mRNA. And so this is the procedure called transcription. And the next procedure is called translation. And in this procedure, we'll get a chain of amino acid. It's kind of a protein, but it's kind of protein. And this procedure happened in the cytoplasm. And I mean, the first procedure, we get mRNA, and mRNA will get out to the nuclear and enter the cytoplasm. And in the cytoplasm, the mRNA will encounter tRNA. I have mentioned these two parts can combine with each other. And another thing is the tRNA also carry amino acid. In other words, this procedure will also carry amino acid together. And the amino acid will hand in hand together to make a chain or protein. So after these procedures, we will get a chain of amino acid. It's a kind of pre-protein, we can say. So now let's come to our problem today. My question is, we have the structure of DNA. We also have this kind of thing, like we also have tRNA. It comes from one part of the deep, as mentioned. This one can also combine to DNA. It's also complementary to each other. So my question is, why we need mRNA? Who can answer it? Any volunteer? Okay, let me give you some hint. As I mentioned before, DNA exists in which place?
Nucleus.
And tRNA exists in which place? The
Cytoplasm.
cytoplasm. So what we need mRNA?
You need a message to send what was
Send the
encoded in
DNA sequence.
Yeah, send what is encoded in DNA to
Don't use encode, it's a little hard.
send the message from DNA to
Yes.
RNA.
We have to send the DNA sequence to the outside nuclear, to the cytoplasm. Here I want to add is DNA can't get out of the nuclear, and tRNA is kind of constricted in the cytoplasm. So we need mRNA. And in other words, mRNA is also called mRNA is short for messenger RNA. And my another question is, since mRNA is short for message RNA, so tRNA is short for what? Can you guess?
Translate.
Let's translate.
Very close.
Transport.
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 Okay, great guess. Transport RNA. Why? Because it carries amino acid, and tRNA can transport amino acid to the sequence and to get the final structure. Okay, after these two procedures, however, we don't have a mature protein. In order to get a mature protein, other procedures are still needed. The amino acid chain may be squeezed or curled together to get the final structure like this. It's just like this kind of necklace be squeezed together. Okay, here is everything about today. Any questions?
No.